Buongiorno a tutti, se qualcuno mi sente.
Okay, here we are finally. Good morning, everybody. And thanks for joining us in this workshop about the In Silico Medicine and diabetes, which is organized by the Italian Medicine Agency in cooperation with the Virtual Physiological Human Institute. Just let me briefly introduce myself. I'm Paolo Foggi. I'm the head of the Innovation and Pharmaceutical Strategy Division at the Italian Medicine Agency. And here with me, surely he doesn't need any introduction, Professor Silvio Garattini, which is founder and chairman of the Mario Negri Institute of Pharmacological Research, with whom I have today the honor to co-chair this event. Before I leave the floor to him, I just want to tell you a few words. This workshop is part of a broader initiative which is aimed at strengthening the collaboration with academia and industry in various fields of innovation. At IFA, as part of the EU regulatory network, and in particular also as part of the EU innovation network, we are working to establish a platform for early dialogue with academics and researchers and industry. We are interested in discussing emerging technology with the aim to identify and promptly address potential legal and regulatory issues that may emerge, identify the need for specialized expertise at an early stage to ensure that here on the regulatory side we get on board all the competencies needed to evaluate new technologies. In the end, regulators should never block innovation because they don't know how to evaluate that. On the other hand, we also believe that dialogue is critical to raise awareness among developers on what are the regulatory requirements that they need to consider early in the development of a new technology. Therefore, I guess that early dialogue means a mutual exchange to ensure the good innovation reaches patients as fast as possible. That's where we are. So today's workshop is part of this effort. I'm grateful to my Director General, Dr. Magrini, for his support on making it possible. Grateful also to all the people who collaborated and made this event possible. One last thing before we start. Just let me remember with you Dr. Giuseppe Pimpinella. Pino is our beloved IFA colleague who passed away a few days ago. I just want to say that we miss him a lot, and he's always been an example for all of us. So I wanted to remember him with you. So Professor Garattini, I hand over to you. Just a recommendation to the speakers, please, to respect the assigned time, because we have a busy agenda in a few hours that we have at our disposal. And Professor Magrini will come later on, so he's not coming here. Please.
Good morning to everybody. I was very pleased to accept this invitation, mostly to learn, because this is a field that is becoming more and more important. It represents, in a way, the future. And today is applied to a disease which is very important, also from the point of view of public health. It's enough to say that between type 1 and type 2 diabetes, there are about 500 million people in the world, and with the large majority of type 2 diabetes, of course. And in Italy, we calculate that 5.8% of the population is affected by diabetes, and which means about 1 out of 18 for the general population, but 1 out of 6 diabetes type 2 for people that are older than 65 years. So it's a very important disease, which is avoidable, and that's an object of reflection, because we should try to avoid prevention. Is not very popular today, but it's the way that we have to go for the future. It's also a disease that is diagnosed. There is one diagnosis of type 2 diabetes each 2 seconds in Italy. So it tells you how important it is. And of course, we are very interested in diabetes because of the important complications that are coming out from this disease. So let's start the program. The first speaker is, just a second.
We have some introductory remarks.
There are some introductory remarks from Professor Del Prato, yes. Please, I believe it's in remote.
Yes, please. He's professor
I'm
of
connected, and I hope you can
yes,
hear me.
Yes,
okay.
yes, very well.
Okay, all right. Good morning, Professor Galatini. Good morning, Professor Foggi, dear colleagues and friends. Thank you very much for this kind invitation. In my position of the current president of the European Association for the Study of Diabetes, it is a great opportunity for me to be here. In particular because, as I can see from the program, it is diabetes that will be the focus of the discussion, exploring the different opportunities that the In Silico Medicine may offer in assessing new treatments as well as in supporting the management of the disease. Now, the EASD is not directly involved in specific actions for implementation of In Silico Medicine in diabetes, besides, of course, fostering and supporting any scientific research activity that may be developed around this growing field. However, the EASD has been seminal in establishing what is now an independent body, which is known as the European Diabetes Forum, which I'm also the honor to be the vice president of. The UDF incorporates various international organizations, including those of general practitioners, diabetes nurses, and of course, those of people with the diabetes, having the goal to ensure that policy action can be directed toward optimal diabetes care. Now, within the UDF, 3 main strategic forums have been identified in order to establish some action. And these 3 forums are data and registries, technology and digitalization, and integrated care. Working groups have been established and recommendations published, and I invite all of you who may be an interest to visit the website at eudf.org so we can see this recommendation. Now, these 3 main areas of interest of the UDF have been made, as you can see, easily take advantage of the In Silico Medicine. Take, for instance, technology. Technologies are driving significant changes in healthcare, providing solutions to assist in preventing, diagnosis, and treating chronic diseases. And diabetes is the champion, if you allow me to say so, of the chronic diseases, while supporting the efficient allocation of staff and financial resources within constrained healthcare systems. Above all, the digital technologies can also help empowering patients to have more control and ability to manage their condition. Digitalization can facilitate the flow of data and can support decision-making by generating, storing, and leveraging this information with the aid of artificial intelligence and machine learning, and this will be discussed in the rest of the day. Digital tools can give healthcare providers an extensive overview of a patient health and health records, making it easier to make informed choices about treatments and care. Digitalization also can lead to more informed decision-making by the healthcare providers, who can be able to access the information from repositories and registries, as well as from apps used by people living with diabetes. The new technologies are indeed opening the door to remote monitoring for disease management by clinicians and improving patient self-sufficiency. Healthcare systems were already under enormous pressure even before the pandemic, but workforce shortages coupled with an aging population are likely to cause further strains in managing the inflow of diabetes patients. Digital tools have the potential to enhance and to enable healthcare system to save time and allocate resources more effectively. And individual data from different resources also give the opportunity to individually tailor treatment pathways, securing the best possible efficiency for the whole healthcare and social system. This concept also opens up to another aspect the ESD is actually working on, that is the precision diabetes medicine. In collaboration with the American Diabetes Association, a working group has been established to generate consensus on the viability and potential implementation of precision medicine for diagnosis, prevention, and treatment of diabetes through expert collaboration, stakeholder engagement, and systematic evaluation of available evidence. The vision of the project is to realize a future of longer, healthier lives for people with diabetes, achieved by applying the appropriate treatment for the appropriate person at the appropriate time. The first document of the working group has been already published in 2020 in Diabetology and Diabetes Care, and in that paper, a roadmap has been already planned. And at the end of that plan, in 5-6 year time, we expect educational initiatives directed to the healthcare providers, as well as to the persons with diabetes, about the implementation of a precision diabetes medicine to be started, and guideline also to be modified to start incorporating new recommendations in precision medicine. Now, a key aspect of the process includes enhancing use of data, and when I'm saying data, I refer to omic assays, electronic health records, patient registries, digital imaging technologies, and or wearable devices. In other words, all information relevant for people with diabetes. And as you can easily appreciate, here the role of in silico medicine may become determinant in analyzing a large bulk of data, in testing precision medicine strategies, in evaluating outcomes and sustainability, and validating modern management of diabetes. It is for all these reasons that I eagerly wait to learn more during the rest of the morning, and I really take the opportunity to thank you for the invitation, but even more for this important opportunity and for really opening up here also in our country what could be the future for treatment of diabetes. Thank you very much for this invitation again.
Thank you for your thank you for your important remarks. I think we can start now the scientific program by introducing Professor Viceconti, Marco Viceconti, who is full professor of computational biomechanics in the Department of Industrial Engineering of the Alma Mater Studiorum, University of Bologna, and is also the director of the Medical Technology Lab at the Rizzoli Institute. Professor Viceconti is the founder and he led for 7 years the prestigious Institute for In Silico Medicine in Sheffield, UK. And I'm very pleased to give you the
Thank you, Professor Gerattini. Good morning, everyone. Okay. I've been already introduced, but the particular hat I wear today is that of a coordinator of a large European funded project called In Silico World that aims to reduce the barrier toward the adoption of in silico medicine technology, in particular what we call in silico trials, so the use of computer modeling and simulation in the development of medical products. I need to, being the first speaker, I need to provide the definition of in silico medicine. As you know, living organism can be investigated in vitro, in vivo, and now using computer modeling and simulation. And the term we use is in silico because of the silicon that makes the computer chips in the computers. What is the situation of in silico medicine in Italy? Well, it's much better than our average presence in research, and especially in technological research. To give you a number, the first road mapping exercise we did at the European level for this topic was a document called Seeding the Europhysium that was published in 2007. and hundreds of experts from all over the world contributed to this document, but 16% of them were Italian. So already at the beginning of this new area, the presence of Italian researchers was very important. Today we celebrate the fact that one of the first in silico technology based on virtual populations accepted by a major regulator as a replacement of animal experimentation in 2008 was developed in Italy. Today we have internationally recognized research groups in the area of in silico medicine in Padova, in Milano, in Bologna, Pavia, Roma, Catania, Torino, Genova. The list is very long and is constantly growing. And another index of our presence is how many projects in the area of in silico medicine funded by the European Commission are coordinated by an Italian institution. And the list has been very long, starting from the 7th framework program when this topic was first introduced. So what is the story so far? Some time ago, the National Health Council, il Consiglio Superiore di Sanità, which is the Italian technical and scientific advisory body of the Ministry of Health, invited Professor Cobelli to establish a working group on in silico medicine. Claudio asked me to co-chair this group, and together we formed this working group that worked for about a year, and in 2020 we submitted to the attention of the minister a report entitled Adoption Pathway for in silico medicine in Italy. Unfortunately, the COVID and pandemics delayed the publication. The document was published only 1 year after, in 2021. But soon after, we started to have a conversation with some colleagues in IFA, and we agreed that there was a potential for collaboration around this new area of in silico methodology for drug development. So together, an initiative we agreed was to develop today this workshop, okay? Why we chose diabetes? As you know, in silico medicine applied in virtually any disease. Why diabetes? Diabetes mellitus is a complex, multi-organ, multi-scale disease for which, however, good quantitative mechanistic understanding is available. And that is very important because, especially for the type of modeling that we are discussing today, which are primarily mechanistic, patient-specific models, having a good mechanistic knowledge of the physiology and the pathology is essential. I already mentioned that diabetes was one of the first target areas that was recognized in terms of in silico medicine tools in regulatory world. And Professor Garantini already explained in a much more informed way than me why diabetes is so important socially. I just add a number. In Italy, we spend around 8 billion euro per year around the care of diabetes. So it's a huge pandemic. It's a huge social and sanitary problem. This is the list of the names that contributed to this work group. As you see, a lot of experts from different institutions with different backgrounds. We had bioengineers, biologists, entrepreneurs, experts of legal aspects, and so on. So a variety of expertise that were brought within this work group and then worked together to the production of this document. The document is available online. You can download it. It is in Italian because it's targeted for our internal use. But I will try to summarize this document in 3 slides by reporting you the 7 recommendations that are at the end of this document. The first 2 are related to the difficulty that the legal system in Italy posed to the secondary use of clinical data for research purposes. I'm sure that most of you are familiar with the specific issue of our legislation on this. We made 2 recommendations in this regard because this is, in our opinion, an essential step to unlock the potential of in silico medicine in Italy. In silico medicine can be used to do a lot of things, but in Italy we think that we should focus primarily on solutions that reduce the geographic and wealth heterogeneity of the access and quality of care, and to reduce the cost of research and development in new treatments, but also, of course, to reduce animal experimentation, which is becoming an ethical imperative in our society. There are 2 recommendations around the funding of particular targeted area in silico medicine. And then there are 2 more technical recommendations related to the data interoperability, especially in clinical hospital healthcare system, which is still very poor in the majority of the country. In conclusion, we believe there is a need to promote the development of best practice to improve the adoption of in silico methodology. So as In Silico World Consortium, we're driving a number of initiatives at the European and national levels, including this workshop of today. I think that there is a special opportunity for Italian biomedical industry. Most of our companies are small or medium-sized, and because of this, they are more agile, they have a higher propensity to adopt more radical innovations. So this is a huge opportunity for the industry of our country. But of course, this won't happen if we don't start an early dialogue between regulatory authorities, academia, and industry. And we believe that this workshop is the first step in Italy in this direction. Thank you very much.
Okay, thanks Marco. You summarized very well, and it's a very interesting one to introduce and to set the scene. Now I call here Claudio Cobelli, Professor Cobelli, which is Emeritus Professor of Bioengineering at the University of Padova, and is renowned for his research activity in modeling and technology of diabetes, and it's going to tell us about the FDA-accepted simulator. Thank you. Please.
Professor Garattini, Dr. Foggi, thanks Marco for the nice setup of the In Silico Medicine in Italy. The title is the FDA-accepted UVA Padova Type 1 Diabetes Simulator, some ideas, methods, clinical applications. This, for us, was really a great news. So in January 2008, the FDA accepted the simulator as a tool for preclinical studies, so in order to skip animal trials. As you can see, some details below. We won a contest with other academic group and other companies. But what I would like to stress is that the situation, the context was really ripe. So in the sense that sensors were becoming accurate and more accurate and more accurate. So this means that, as you know, garbage in, garbage out. If you have a good signal, you can probably close the loop. So this was the idea. And then JDRF was really lobbying for accelerating the artificial pancreas research. What was happening before the simulator? You have to do animal studies first and then humans. And this is a classical example, a prototype of Medtronic. They did first in 8, I don't know why they call it canine animals, but 8 diabetic dogs. And then they went into humans with 10 patients. You can argue what you can learn from 8 diabetic dogs in terms of variability. Probably nothing. But this was the situation. The idea was that we substitute animal trials with in silico trials, and there is no point on going into details, save years, money, etc. To do this, we needed a new generation of simulation model, because clearly you have to be very accurate. So you have to have a large-scale model, which we call maximal. You have to describe the population variability. You will see in a slide, next slide, the variability which is there in the normal population, not mentioning the type 1 diabetic population. And clearly these are mathematically nonlinear, high order, large number of parameters. What was the breakthrough to get to this new simulation model? This was a triple tracer meal studies done at Mayo Clinic in Rochester, Minnesota. I'm not going into the details of the triple tracer study, but the point that you have to remember that thanks to this technique, which is very complex, and clearly it is not by chance that it was done at Mayo Clinic, you can measure independently, virtually model independently, in addition to concentration, which you are measuring, fluxes, which are not directly measurable. So, for instance, we did, there are a database of 244 healthy and type 1 diabetes subjects. And just to give you an example, if you have only the concentration, you have these 2 time courses during a meal, glucose and insulin. And you can see already the variability there. But now, with this technique, you have the fluxes. You have glucose production, you have rate of appearance of glucose during the meal, you have glucose utilization, insulin secretion in a normal subject, just an example. Look at the variability. This is the normal population. This is the structure of the model. So you have below the insulin system, just in a healthy individual, just to give you an example. Then you have the glucose system, gastrointestinal tract, and so on. This dashed line with the bullet means where you can measure. In addition to measure concentration, you can see that you are measuring also fluxes, glucose production, glucose utilization, secretion, rate of appearance of the meal. It has been due to this unique and rich database that we were able to identify in each individual that model by technique which some of you know better than me, Bayesian forcing function strategies. The results has been this UVA Padova, because forgot to remember that the work was done with some colleagues at the University of Virginia. So this is the simulator. We created 3 populations, virtual population, adults, adolescents, and children. And this is just an example of the variability of insulin absorption, insulin action. So these are distribution where all these 244 individuals lie. We used the simulator for artificial pancreas research, for designing the controller, testing, and also for in silico trials for regulatory purposes. Just to give you a very clear example of the importance of in silico trials, the simulator was accepted by FDA in January 18, and we did the first experiment at UVA and in Padova in April 17 and May 20. So after 3 months, we were able to go in the hospital, obviously, not free conditions, but 3 months. So no need of dogs, no need of laboratory, simply an in silico strategy. We established also a very nice procedure with JDRF. So this was a consortium of investigators, a JDRF Artificial Pancreas Consortium. So if an investigator has an idea of a controller, he sends his controller to this Jeff Center in Florida, where the simulator is housed, because we want to be out of the loop. They do all the virtual trials, send the results to FDA, and FDA can say yes, no, revise. And out of the story, there were 140 candidate algorithms version which has been submitted to FDA. Out of this, only 16 projects were accepted, and they got an investigational device exemption in order to start human studies. The simulator was not only used for artificial pancreas research, but we have also used it for a variety of important technologies, like glucose sensors, new molecules, and new insulin delivery modes. I'm not going into the details of all of them, just a few words, and then some more words for some of them. The very important is this idea of the glucose sensor. So before this approval of FDA, if you are wearing a subcutaneous glucose sensor, in order to dose insulin, you have to do a blood test, a finger prick. After 2016, the sensor, the signal of the sensor can be used to dose insulin. You can imagine, the big step forward to this. This is called non-adjunctive use. So you don't need to do self-monitoring of blood glucose to dose insulin. At that time, Dexcom didn't have enough data to go to FDA. So they asked us to do massive simulations in order to complement their data. The simulations were really quite a number. At the end of the story, they were more than 40,000. 40,000 simulation. Oops. Okay, I told you that there were 40,000. And this were, the FDA, it was voted, 7 against 2, it passed. So since 2016, thanks to the simulator, there is this non-adjunctive use of glucose sensor. We have investigated also adjunctive T1D treatment with pramlintide. Pramlintide delays glucose absorption. So with the subcutaneous, which is delayed, the two time courses of glucose and insulin are more matched. So simplifies a little bit. You can see here first an in silico paper, and then followed by a clinical study. Very interesting also was the inhaled insulin, Afrezza. Just to give you, because I like this slide, so you can see that you have the insulin, inhaled insulin, which entered the simulator. Clearly, think of the difference between inhaling insulin and subcutaneous insulin. Dramatic difference in terms of the time courses. Inhaled insulin is very fast. So how can I do the dosing? In fact, you do either post-meal or split dose during the meal. So this helped a lot, the assessing the Afrezza insulin. We have investigated also the new basal insulin, Glargine and Degludec. But I would like to finish with this last insulin delivery through the peritoneum, which is part of my current research activity, with this Forget Diabetes project. Forget Diabetes is clearly the message is there. So we want to do something that people can forget the disease. This was a future emerging technology call. As you may know, these are very competitive call. It's almost science fiction, but with some good background. And in fact, they call it high risk, high gain. So nobody knows if we will arrive at something useful, but they gave us quite a lot of money for millions for 5 years. And we have 7 centers in Europe, 2 Italian university, one is the Scuola Superiore Sant'Anna in Pisa, and then two German institutions, 1 French, a new sensor done in Norway, and so on. The idea is to do a sort of bionic implantable pancreas, which has a glucose sensor in the peritoneum and the infusion of insulin in the peritoneum, and then a control algorithm. This is the idea. The pump is very innovative. I'm not going into the details of that. Intraperitoneal, fully automated closed-loop studies are very difficult, very demanding for the patient, and very demanding for the investigational team. So they are usually, this is not by chance that this is the first closed-loop study with intraperitoneal delivery. 1 day in the hospital, this was done in Montpellier, with unannounced meal. This is one major limitation of subcutaneous insulin delivery, because you have to announce the meal. So you have to insert in the computer, even in the 2 commercial systems that are now FDA marked or CA marked, you have to put in the amount of carb. This study was done in 10 individuals, 1 day, because this is really demanding. So it is not that you can take patients and let them stay in the hospital for a long term. So in silico strategy was very useful because we augmented the clinical trial. These were 10 patients, and based on that, we were able to augment the clinical trial and use 100 in silico subjects based on these 10 patients, and do all the simulation that we wanted to do in terms of this picture. It may be a little bit complicated, but focus on the blue line and the red line. This is 1 day, average over 2 weeks. So you can see that with the blue line, you are within the target zone with a very small variability compared to subcutaneous. And this is unannounced versus announced. So this is very important. I think that maybe I'll skip this table because you may go sleeping. So I think that the major messages, I think, came through. And here are some conclusions. So the acceptance of the type 1 diabetes simulator was an unprecedented event in the modeling community. It was really something that incredibly novel. In silico trials have dramatically accelerated regulatory processes of artificial pancreas research. I have shown you example where the simulator has been instrumental in various diabetes technology research lines: glucose sensor, new molecules, pramlintide, Afrezza, and insulin delivery modes, intraperitoneal versus subcutaneous. In silico trials allow augmentation. This, I think, is a very important point. Augmentation of small clinical trial by increasing numerosity and variability of the population. This is typical in the intraperitoneal. You have 10 patients, small variability, but what will happen when we have 100 subjects doing the same job? And the last is a negative statement. Absence in Italy of the FDA investigational exemption device penalizes research and especially innovative biotech companies or investigator-initiated studies. This is really a very negative point that I'm not the person to solve it, but probably in this room there are persons that can help. We don't need papers, we need law to establish this kind of things, because I can tell you my experience. We have to return to NIH $200,000 because we were not allowed by the Ministry of Health to do experimentation. All the money went to France because they can do in 1 month an investigational exemption device. Recently, the university, the Bocconi, did some work in collaboration with the Ministry of Health, came out a paper, but a paper is a paper. So you don't go far with the paper. We have to implement these strategies in practice. Thank you.
Thank you, Dr. Cobelli, for this interesting presentation. I'm sure that this symposium will be of help to cancel your negative statement, and I think that there is all the goodwill, I believe, from IFA to do something in this respect, and that's also the reason why IFA has co-sponsored this kind of meeting. The next speaking will be Chiara Dallamanna. She's professor of, she's part of the biomedical engineering from the University of Padua, and she is associate professor of bioengineering at the University of Padua always. Okay. The title of her presentation will deal with the type 2 diabetes instead than type 1. Please.
Thank you for the presentation, and thank you for the invitation to this workshop, which gives me the opportunity to talk about the type 2 diabetes simulator that has been developed by our group in Padua University. No? Okay, so I think it is of great importance because type 2 diabetes is much more widespread than type 1. Almost 90% of the subjects with diabetes have type 2 diabetes, and so the fact that we can run in silico trials in type 2 diabetes is very important, because also in type 2 diabetes there is this need to accelerate the development of new anti-diabetic drug and treatment. And so my aim here is to present this type 2 diabetes simulator that we will see is able to reproduce the variability of that is observed in type 2 diabetes population, which is huge. But I also will show you that this tool can be used to experiment in possibly rare pathological conditions, so in very extreme pathological conditions. I don't know where I have to. It's not working. Okay, so I'm sorry. I don't know where I have to. Oh, there. Okay. Sorry. So just to recall you, what is the structure of a simulator? from our point of view. A simulator is basically a set of model equations, so there is some math behind the simulator, and a set of virtual subjects. We have already heard this definition in the previous presentation. What is a virtual subject from the mathematical point of view is a vector of model parameters which makes the model behave in a different way, and so to resemble what can happen in a real subject. So each subject is responding to a stimulus in a different way, and this is what we want to replicate in silico. And then there should be an infrastructure that usually is a user interface that allows people to set up the simulation scenarios, for example, the duration of the simulation, the time and amount of the meal, possible hypo rescue, or some scheduling of the drug delivery, and so on. This is the scheme of the model on which the type 2 diabetes simulator is based, and is similar to what Professor Cobelli presented, but the difference is that the type 2 diabetes subject still secrete insulin endogenously. So there are 14 differential equations and 39 parameters to describe this model, and at variance with the type 1 simulator, we have to describe also the beta cell function and the extraction of insulin by the liver mainly. And this allows to simulate not only plasma glucose, but also plasma insulin and plasma C-peptide concentration, that are the measurements that usually are taken from subject to have a clear metabolic portrait of a real subject. To identify and tune this simulator, we use data coming from 2 studies, both made at Mayo Clinic, with the triple tracer protocol that Professor Cobello mentioned, which, and they have been performed on 51 type 2 early stage subject. Early stage, I mean subjects are not taking insulin. So they have still their own endogenous secretion. And so, in addition to plasma glucose, plasma insulin, and C-peptide concentration, we had access also to the fluxes that Professor Cobelli was mentioning. So, endogenous glucose production, the meal rate of appearance, and glucose utilization in this case. And this was very important because we could use this data to identify this model, so to put number on the parameters of the model, because the model is very complex, and so we needed a very large data set with all this information on the fluxes to be able to identify it. This forcing function strategy is represented in this slide. So we basically divided the model in sub-models, subsystems. Each of them is perturbed by some input and produce some output, and they are all interconnected to each other. So this forcing function strategies works in this way. Let's consider, for example, the subsystem with muscle and adipose tissue. So in this case, this is a scheme of the model. We use some of these variables that we have measured or estimated, these fluxes, like insulin, endogenous glucose production, and meal rate of appearance, which represent, in this case, the input of the model, and the output of the model, in this case, are plasma glucose and glucose utilization. Using this procedure, we were able to estimate the parameter of this sub-model, and we repeated this procedure for all the subjects and for all the subsystems. So at the end, we came up with a number of model parameters, of parameter vector, in this case 51, that completely describe the system. And from this information, we were able to build what is called the n-dimensional distribution of model parameter. What is this? This is a mathematical object that represents the probability that a given combination of parameters can occur in a real subject. Okay, so when the value of this multidimensional function is high, means that that combination is likely to occur. If it is low, it means that it is a rare subject. Okay? And so from this distribution, we were able to generate the desired number of virtual subjects. In our example, we usually use 100. It is a sort of magic number, but you can imagine that it is a sort of compromise of having a manageable number of subjects then to analyze, and a substantial number on which drawing our conclusion. And as you can appreciate from this slide, each parameter vector makes, in this case, glucose curve change under the same stimulus. So we created virtually the variability of the population. And if we focus, if we focus on two very important parameters that are insulin sensitivity and beta cell responsivity to glucose, we can appreciate that in this population of type 2 diabetes subject, we have that both insulin sensitivity and beta cell responsivity are, on average, lower than the same parameters estimated in a healthy subject, the red curve versus the blue curve. Okay. We validated this approach and compare the simulated curve of glucose, insulin, and C-peptide with the real one. And you can appreciate from the upper figure that both the average and the variability of plasma glucose, plasma insulin, and plasma C-peptide are similar in silico and in vivo, and we were able to well cover both the average and the variability. This was the most important thing. So this validated our methodology of extracting the subject. But as I promised with the title, we said that also this methodology can be used to test the efficacy of a given treatment in rare but not so rare subjects. So what does it mean, this? It means that we have to find subjects, or we can find subjects that are not very likely to occur, but they can occur in practice. They are rare but not implausible, and we can find this subject in the tail of the distribution. For example, this is the first rare subject number 1, which in the example has very low insulin sensitivity and pretty high beta cell responsivity. So it's a subject that is not very common, but it can happen with some probability, even if this is small. There is another example, if I can have it here. Oops. Subject number 2, the yellow one, which conversely has pretty high insulin sensitivity but pretty low beta cell function. Or the orange one, rare subject number 3, which has both insulin sensitivity and beta cell responsivity, which is really impaired. Okay, so the message is that we can focus on the tail of the distribution and assess our treatment or our drug, or, for example, decide which is the best dosing regimen for such population. And in fact, we ran, for example, just to do an example, an in silico trial in these rare subjects, and we selected 10 subjects with severe impairment in both insulin sensitivity and beta cell function. So like the rare subject number 3, and we want to answer this question if this subset of subjects can benefit from having a double dose compared to a standard dose of a given drug, which is designed to improve both parameters, insulin sensitivity and beta cell function. So what we did was to run an in silico trial first in the 100 in silico patient, which received the single dose of a drug, so 1 pill per day, for example. Then we ran the same trial in these rare subjects, again single dose of the drug, and we tried also to give virtually to the same 10 subjects a double dose. What we found was that actually our 10 rare subjects really had benefit of taking a double dose. And in fact, the outcome that in this case was represented by the area under glucose curve was more similar to the average population receiving 1 single dose if they receive a double dose. So this means that sometimes looking at the whole population with the 100 subjects having the single dose, perhaps it seems that on average they are performing well. But perhaps it's not the right thing for all the subjects of the population because there is a lot of variability. So sometimes it could be of great importance to be able to simulate what happened in these rare subjects. And this was obviously just one example of possible use of the simulator. And to conclude, I can say that our simulator that contain at the moment 100 virtual in silico subjects could be of great value because it can help in running in silico trial in a large population like we usually do in phase 2 clinical trial. The fact that in silico we can control and minimize the intra-subject variability is also a great thing because we can implement sort of ideal crossover design, and so we can really see what is the effect of a drug or a treatment. And as I said, this possibility to, let's say, virtually recruit a subset of subjects with common characteristics, for example, low insulin sensitivity and very low beta cell function, can help in design the best dosing regimen in this such population. And so this can help in possibly improving diabetes therapy in these rare, but as I said, not so rare subjects. And these are the acknowledgements because all this work has been done in team, obviously, with my colleagues from Padova, and it has been possible thanks to the availability of the data which came from Mayo Clinic and the University of Virginia, and I thank you all for your attention.
Okay, many thanks, colleagues, for this very, very interesting presentation that we had. And we decided, it's the privilege of the chair, to skip the discussion right now, to have the discussion at the end of the presentation, because I guess that we can have many additional, very interesting inputs. So I kindly invite Dr. Castiglione. He is a computer scientist and is a research director at the National Research Council of Italy, and is going to talk about the potential predictors of type 2 diabetes risk, machine learning, synthetic data, and wearable health devices. Please.
Good morning, everyone. Thank you, Professor Gattini, Professor Foggi, and Professor Marco Viceconti for having invited me and give me the opportunity to tell you what we have done, the mask, yes, what we have done in a European project. So the work we have done is well aligned with this workshop. So we basically, the starting point, we're talking about type 2 diabetes here, so not type 1 diabetes. So Professor Dallaman has very well introduced the problem. The approach we follow, though, is not that of the treatment of type 2 diabetes, but that of prevention. So the model is somehow different. And it starts by, let's say, saying that type 2 diabetes, there are evidence that it's an autoinflammatory disease, two evidence. One is that there are inflammatory markers in type 2 diabetes people, and also that if one uses anti-inflammatory drugs, the patient has a benefit. So there is a low-grade inflammation going on, which make the people going from normal or healthy to sick patients. So the model has been built in Mission Type 2 T2D project. It's a FP7 European project, concluded already some few years ago. And the goal of the project itself was to build a computational model. So we built a mathematical computational model which wanted to take into account lifestyle patterns of an individual. So since we are talking about pre-diabetic condition, and we wanted to trace the trajectory of the individual from normal to, let's say, at higher risk of type 2 diabetes, so not yet diabetic. So we took in consideration variables describing the energy intake and variables describing the physical activity pattern. And we converted this into do we have a laser? Yes. Into a number which basically gave a score of the risk of type 2 diabetes. And this was done through dynamic modeling of a number of variables describing the metabolism and the immune activation, so the inflammation. This is the overall picture. If we look at the block diagram of the model, I'm not going into the details, we see that there are quite a lot of interconnection between components. The model itself is composed by 6 submodels, all available in the literature. We didn't explicitly code anything new. We just assembled pieces, and it has been a challenging task that already. And so what we did was basically to code in the input the, let's say, anthropometric characteristic of a virtual patient, so describing the age, the body mass index, and so on. But not only that, but importantly, we also coded for the lifestyle pattern. So, for example, on a time scale of a week, how many times these people used to go to the gym or exercise, for how long, and with which intensity. Of course, very rough. I mean, the idea is it goes beyond that, but at the beginning it was like discretize the space of possible activity levels, discretize the space of possible nutrient intake in terms of protein, fat, and carbohydrates. So this vector goes into this model, which is composed by a module for the inflammation. It's an agent-based model for those who make modeling, which describes how the immune system responds when fat-derived peptides are exposed by hypertrophic adipocytes. So basically, when the adipose tissue is too expanded too much, there is a more likelihood to present this. Probably this is not the only cause, but it's certainly something observed in reality. And it links the increase of weight or increase of the adipocyte tissue with an increase of inflammation. Then we have another model which codes for the metabolism during the exercise, physical exercise. And this model includes also a glucose, glucagon insulin control. Then we have food intake and gastric emptying model from Professor Cobelli and Dalaman, which are here present. And this one has been plugged into the M2 model. Then we also plugged another model, which is coding for the energy balance and gain or loss of adipocyte tissue. Then we also coded for another model linking the physical activity and IL-6 release from muscle cells. IL-6 is one inflammatory cytokine, so it is important to take it into account. And finally, but I have to say that this is has been less used in our study, but we also try to link the microbiota composition with an inflammation through a model called the multiplex network, which is meant to basically create a link between the composition of the microbiota, or the dominance, actually is the production of body rate, basically, of the microbiota with the risk, with the propensity to have a pro-inflammatory immune response, basically. So very stylized, simple, but it's there. So all this model basically creates trajectories in the space of the variables, trajectories in time. So we can, giving an input, what is the lifestyle pattern of a virtual individual, the model gives the kinetics of all these variables, and we look at that output to understand if the trajectory is going toward a higher risk of type 2 diabetes or a lower risk of type 2 diabetes, so quite direct. And so if we look at simulating, when we simulate one day, we look at this kind of plot, we see the relationship between insulin and glucose, and it's quite consistent with what expected. And what is nice is the fact that we also have a decrease due to, in this light blue area, the individual does a physical activity, and that's why it goes down. So it is one of the few models which couple both meal and physical activity. Now, if this pattern we have seen in the previous slide of one day is repeated constantly, so without changes for a long period of time, that's what we wanted to observe. And we have simulation like this. For example, a person which does a long period of high caloric intake, and then it changes mind. He understands that it's better to start doing something for himself and reduce the caloric intake and start to do physical activity. So this plot shows the intensity and where in time this individual has done physical activity. This plot here shows the, let's say, average level of protein, carbs, and fat assumed daily. Okay, so the model in this case shows this kind of trajectories. The variables is a large number, but here we plot only insulin and glucagon, which on average follow this pattern. But what is more important is the fasting glucose, which is at risk at a certain point to go from the green area, which is normal or not at risk, to the yellow area, which means that this individual was going, of course, it was progressing toward high risk of type 2 diabetes, but it inverts the direction in the last month because he exercised and eat less. Very simple. So the model has been validated with the literature. We have validated the model with the data from physical activity only, or food intake only, or food and physical activity combined. What it is less validated, because it's difficult to find the data, is the inflammatory aspects, because it takes time to develop the inflammation. In fact, we had data from a study called the Whitehead, which used to challenge individual for 2 months, I believe, and with high calorie challenge of 2 months, and in these 2 months the inflammation doesn't just appear. So it is clear that there is no such data available in the literature. It's quite difficult to make an assessment of what happened in terms of months or years, because these are the kind of things that the model can do. So we had to somehow rely on previous study of the use of the immune system response in general. So the other thing we have done, since the model itself is quite heavy, it takes sometimes a day to simulate 6 months, it is something that cannot be executed on a cell phone or whatever, a portable device. But we wanted to go into the direction of providing some self-assessment tool. And most importantly, we wanted to measure the lifestyle directly through wearable devices. So not giving a stylized average, pattern at the week level, like I go 3 times and I exercise for an hour. We wanted to have a real-time measurement of these things. So we wanted to connect this up with the real-time monitor. So we realized this partner Medisana realized that this app, it's called VitaDoc, actually is not, it has realized the module inside their app, which is called VitaDoc Plus. But it has used the output of the model to provide the user with this possibility to do some self-monitoring. And how did we enable a phone with the possibility to run such a complicated model? We had to train a machine learning model, of course, because as we know, machine learning is it's basically statistical learning. It's great. It might be heavy to train a model, computationally speaking, but it's very fast in providing an output once it has been trained. So we ran a large number of simulations, more than 45,000, and this data set was used to feed the machine learning model to train the dynamic of the model. So that the machine learning model was then able to do the same type of prediction the computational model was doing. So giving an input was giving an output in terms of how risky, what is the risk of going to type 2 diabetes. So as we have seen in the previous presentation, we coded the variables of the virtual subject in a vector, so sex, age, body mass index, pattern of physical activity, pattern of food intake, stylized per week. We coded this into a vector, and the model returned the time-dependent variation of some variables. We choose these 3 variables to determine the state of a person. So we choose the body mass index, the glucose-based level, and the TNF as a proxy for the inflammation level of the individual patient. With these 3 variables, we used a machine learning algorithm called random forest, which is basically a generalization of a decision tree. A decision tree is a way to classify points in a multidimensional space by means of hyperplanes, basically. So you separate points in space, simply speaking, and you do it by determining a path, a decision path through the variables. It's a quite simple and nice classification model, and the performance we got were very nice. And as I said before, it runs in terms of milliseconds. So when you give a pattern and input, it responds immediately. The computation would take a day to run, but this one would respond immediately. As a side effect of the use of this machine learning method, we obtained a ranking of the variable importance, which gave us the opportunity to look a little bit into the very complex nonlinear dynamics of the model. And for example, we understood that physical activity is less important than food, than intake in progressing into the type 2. We found that the greatest determinant of the body mass index and the glucose-based level, which is strictly linked to the risk of type 2 diabetes, is the body mass index at time 0, so the initial condition of the person. We also found that glucose-based level is driven by the amount of carbohydrates, mostly, and that inflammation is driven mostly by age, followed by, in order of importance, by the carbohydrates and the body mass index. So this was a kind of insight the model gave. So I'm now here to conclude by saying that we wanted to build a self-assessment tool because everybody knows self-assessment can be very good in increasing the awareness of the people, so in promoting prevention of type 2 diabetes. And we have built a computational model which is quite complex, but we were able to synthesize the dynamics into a machine learning method which can be run on mobile devices. And both the machine learning method but also the computational model can be used to compute optimal lifestyle patterns in general. So not just to define the risk, but also to optimize, in terms, again, of prevention, to optimize the lifestyle of a person in this respect. So I want to thank you for your attention, and let me acknowledge Professor Dr. Palumbo, which is here present, and Paola Stoffi and Paolo Thierry, which are my collaborators at the CNR, and all the partners of the project, which of course contributed. Thank you very much.
Next speaker will be Flora Masuamba Shinanu. I hope that the pronunciation is the correct one. She got a master in pharmaceutical medicine and a PhD in pharmaceutical sciences. She will talk about the modeling and simulation, regulatory challenges and opportunities. Please, I think it's online, she will be online.
Yes, good morning, good afternoon, everyone. I see Paolo knocking his head, so I guess you can hear me. So, but still I am not able to move the slide as I would like to. Do you see my very first slide now?
Yes, now we see the title.
Yes.
Yes, very good. So thank you very much for the invite first, and giving me the opportunity to give the regulatory perspective in this very insightful meeting. So I was asked to discuss with you about, the regulatory experience in assessing different types of models, and also including the models, the type of model that has been discussed here this morning, which are mostly simulation models and apply to the field of diabetes. I will give you an overview on the way we are looking at modeling and simulation, in particular in my experience with the European Medicine Agency. And the rules, as you can imagine, would not change because we are now focusing in the particular case of diabetes. So everything that I would be discussing here would be applicable to diabetes, would be applicable to the kind of simulators that you're using here, but can also be expanded to estimation models and other kind of models and simulation in other diseases. So this is a brief outline of the presentation. and I would be presenting you the different applications that we see of modeling and simulation in regulatory submissions, the guidance document, and also discuss with you the challenges and opportunity when comes regulatory assessment of modeling and simulation. So as an introduction, I need to say that the role of modeling and simulation as increasing in the field of drug development. We see it more that you have as drug sponsors in particular that are increasingly using modeling and simulation. And this also reflects in what we see in the regulatory setting. We see more and more modeling and simulation related application. They don't increase only in number, they also increase in the importance. So historically, we were seeing a lot of modeling and simulation application just to describe the data, sort of supportive evidence, but now it's moving into more key source of evidence, like the most important one, which I will be calling high regulatory impact application, but I will come to that. So, because the models are very variable, because the applications are very variable, as a regulator, we need to find out a way to objectively and fairly look at the different models that are being submitted to us and assess them in a consistent manner. For that, we are acting at different levels. I will be presenting you the different guidance document. I will be presenting you the rationale that we are developing now into risk assessment of the different models, and that would be the basis on discussing the different challenges and opportunities that we can have as regulators. So starting with the challenges, one of the big challenges that we currently have, and that will be acknowledged from both sides. When I say both sides, I see the developer side, the sponsor side. The sponsor can be pharmaceutical industry or consortia developing different types of models. And the other side is the regulators who need to look at this model and give the stamp and to accept that this model is fit for purpose and is acceptable for the claim it is being proposed to. So both sides, we agree that there are some gaps that constitute challenges. These gaps, first of all, are in the standards part. So we are currently not having comprehensive guidance documents to cover all types of model. As I said, one of the challenge with the model is that you don't have one type of model. You have different types of model. You have what we call empirical models that are mostly data-driven, like the one that was presented now on machine learning, etc. We also have empirical models that are developed based on clinical trial data. But we also have more and more these mechanistic models that are based on knowledge, that are based on known mechanisms. They are fed by some sort of a priori knowledge, and these models feed simulators. The guidance that are currently available, at least from EMA perspective, are guidelines on PK model, because historically these are the ones that we have seen first. And so we have reached maturity and level to kind of give some very clear guidance on. But there are also several more complex models for which we don't have clear guideline documents. This is one challenge. And sponsors are really asking us for having clear understanding on the requirement when it comes model planning, development, and evaluation for the different particular models. But as I said, it will be very difficult to have a guidance document on each particular type of model. And the ICH have now adopted a new guidance document that will start in June, which will be a overarching guideline on modeling and simulation. I think this is already a good way to address this challenge. And there we would try to have some sort of guidance applicable to different types of model. But for now, there's a gap there that we need to acknowledge. The other challenge that we are facing now, in particular with the kind of model that we are presented today, that challenge is related to data. Why? Because these models, as I said, are not data-driven primarily. They are knowledge-driven. This means that they are fed based on curation, based on data that are not generated by the team which is generating the model. They take data from the literature, they take data from different various sources, and it's not always easy to establish the validity of this data in terms of quality and also in terms of relevance for the final model. So these are challenges that we currently face. The other type of challenge that we face is very simple. It's also related to terminology. As I said, different types of model, different terms used. When we say validation, I'm not sure that all the modelers going from POP-PK model, PKPD, or In Silico model that were presented this morning, we mean exactly the same thing. So there is also like a sort of communication issue there. We need to agree on some sort of terminology and understand what each different people mean by different terms. And also you also have a lot of technical details that can be presented when sometimes not needed. So the perspectives are different, the terms are different, and the presentation, the technicality are different. All of these represent challenges, and I will be presenting you now the way we kind of operate from EMA side. What do we see and how can we address these different challenges? The first message here is that irrespective of the models, the complexity of the model which is being proposed, from my experience at least, the question, the actual application that the model addresses are very common actually. I have barely seen a sponsor or a consortium coming with a very complicated and completely new application for their model. No, most of the time, these are models for trial design optimization, dose finding, and asking a waiver for a study. It can be an animal study, it can be a human clinical study, or asking a waiver for conducting a different study that could be regulatory mandatory. So these are the kind of application everyone more or less come with. You have variant of, but we are generally in those space. Why am I showing this? This is just to tell you that to us, model is a method. To address this kind of question, there are alternative methods. And the old game would be to show that the model-based claim is not lower quality alternative method that can be used to address exactly the same question. When come regulatory interaction with the different consortia or sponsor, you have the different regulatory procedure. I have copy-paste here on the slide the EMA website. When the different procedures are well explained, you have the contact persons and their email. Take-home message here is, please come to regulators as early as possible. You have a procedure like Innovation Task Force briefing meetings. These are very informal meetings. You can come at each stage, so come early. The advantage of coming early is that you engage the conversation with regulator and you are more open to the comment. When people come very late, they have conducted all the experiment, all the simulation. When the regulator challenge those and say you have to redo the whole thing, the receptivity is much more limited than what we see at very early stage. I have also highlighted the qualification opinion. Why? Because when we have qualification advice or opinion, the discussion with the regulators are at the method level. We are not pressed by a particular drug development program. We are not pressed by any kind of marketing pressure. We are really discussing at method level, and I see an increasing number of consortia coming with their model for qualification advice, qualification opinion. This is something which I like a lot, and I would like to press everyone who has very interesting model here to come to regulators, discuss the context of use and the different application. This is well seen. As I was saying, an example here, just because you were talking about diabetes here, one example here is a scientific advice request that we received a few years ago when the company wanted to very change their initial mind for their pediatric investigation plan. Initially, they were planning to conduct a study in pediatric population, and because they were facing some enrollment issue, in the enrollment of children in the study, they proposed to shift to a model-based approach in order not to have a big study, but have a smaller study in the pediatric population that would be informed by some modeling and simulation exercise. And we did have a very positive and insightful discussion with this sponsor. In terms of guidance document, again, go to the EMA website. Several guidelines are present, are there, Q&A document, concept and reflection paper. Some of those are related to modeling and simulation. They are not legally binding, but they constitute some good basis for discussion with regulators. When comes simulation-based model, the type of model which are called in silico models, I think in silico models should be seen broader, but when I hear in silico models, most of the time, these are this kind of simulation model when you have, some sort of mathematical component behind, and then the variability is actually imputed in the in silico trial component of the model. The guideline which is closer to that type of mechanistic model is the PBPK guideline. In that guideline, because for PBPK, for a physiologically based pharmacokinetics model, it's also not an empirical model. This is kind of mechanistic model. And I think that several components of this guideline would be applicable to the mechanistic model that I have heard about today. And which is normal because, as Marco was saying rightly, diabetes is one of the fields where the understanding has advanced a lot the last year. So you go with mechanistic model, look at the PBPK model, principle should be applicable to this kind of models. Regulatory impact now. The regulatory impact, as I said, is related to the scrutiny that regulator would have in looking at the model. And this is quite intuitive that the more importance the model has in the development program, the more demanding the regulator will be. And we rank the different importance of the model like based on this impact scale. We consider the model as a regulatory impact model when it is proposed sort of to replace an experiment, a clinical study, basically. And for that, we will need to have a very thorough understanding of all the components of the model, and we would really strongly advise to go for a qualification opinion so that we can accept this. For high regulatory impact model, we would look at the assumption in the model, the uncertainty, and the impact of the uncertainty in the model, but also the data, the fitting performance, and also the model structure and identifiability of the different parameters. And of course, we will look at that less for lower regulatory impact. I will finish my presentation on this component, which is one way we are proposing to address the different variability we can have in the different models, and then to ensure also regulatory consistency. As you know there are two ways to look at the model. The first is one I have heard from most of the speakers up to now, which is like using the model to learn, to build up a knowledge backbone. This is part of what is called model-informed development. So you use the model to understand the disease or to understand the drug effect, and then you can use that model after that to predict yet an observed situation. But as a regulator, we think that we are most sitting on the position of people to assess the model as a method to answer a particular question. So the starting point for looking at the model is the question. And there is a framework that was proposed initially by the ASME-VNV for medical devices that is being adapted now for drug assessment that I believe gives a very objective manner to look at the different models. So the framework is very simple, actually. It's saying that you have a question, you have a context of use, you as a model developer need to first establish the acceptability criteria for your model given the context of use and given the data you have. After that, you need to look at the regulatory impact, the risk of the model, what happens if your model is wrong for the patient, and then produce the result of the acceptability criteria and come with the answer to your question. Like I said, the question, the application are the same. The model can vary, but the criteria to look at the model need also to be very consistent. Several papers have been published on the application of this credibility-based approach on drug development and on model for drug development. I don't have time to go into that in details now. I can only recommend you strongly to look at this. This is the matrix that you will use, you will see in these different publications. I strongly recommend to use it because it's very simple. It's also addressing, the communication issue that we do have with the developer of the model. So very clearly state what is your model, what is the question the model is addressing, what is the context of use, how do you yourself assess your model, what is the regulatory impact, what are the risks, and what is the decision based on the result you obtained. Data is important, not only where you found the data to feed your model parameters, but also are these data relevant for the simulation you are trying to do with your model. And for that, I can only refer to what is said in the PBPK guideline, very relevant for all kinds of mechanistic models. And this is another table, again, in this publication related to risk-based assessment, where you really have explicit component where the regulator will look at to assess all kinds of models, including the very complex model. As you can see, we don't look only at clinical validation. We also look at technical validation. The way the model is implemented, the way the computation are performed, all of these are part of the qualification process. So I will end in when I started for the credibility assessment. There's a question. The modeling and simulation is one method. Think about the alternative method. Think about the evidentiary standard for that type of question. Model should not give less than what a clinical study would give. So, and regulators are there to ensure that the model accepting modeling is not giving in. So, as I said, models have improved in importance in drug development, and also the regulatory assessment is maturing over the years. Based on experience also, we are also seeing from the different applications. Modeling and simulation is a method. There are alternative methods. Modeling and simulation is a method to address a question. So it cannot do less than the alternative method, otherwise regulator will ask for the better than modeling methods. And we recommend, we support the risk-informed credibility framework because it increased transparency, communication in what the regulator can be expecting for the model. regulatory impact is important, data quality is important in the communication with the different regulators. Thank you for your attention.
Many thanks, Flora, for this very comprehensive presentation, and we want to hear the regulatory perspective, but it is quite important. Now, the floor to Jim Bosley is going to present the GINCO platform, is the voice of industry here. Please, Jim Bosley.
Okay. Can you hear me? Please answer with the
I
audio.
kindly recommend to stick to the time assigned, please, so that then we can have
so I have
discussion.
6 minutes, or I have 15 minutes. Can you hear me?
15 minutes.
Okay, very good. So I'm going to go full screen here, and let me know if you can't see the. Let's see if I can do this. Advanced. I am going to go to. I'm just going to do this then. I think that's. Can you see the slides here?
Yes,
Yeah,
not
very
full
good.
screen, but.
Let's see if I can do this.
Okay.
Let's see. Is that full screen now? Yes? No?
Not yet. But.
Okay, this is the best I can do. I tried to go full screen.
Okay.
on
Anyway,
the-
we see. No problem.
All right. So I'm going to be talking about a platform model. First of all, I want to thank the previous speakers. I found everybody's talk great. I thought Flora's thoroughness in assessing model credibility is very useful. I will say this, in industry, actually, I think there's somewhat of a lack of rigor in thinking about models, and they would do well to look through Flora's slides. Dr. Musamba, sorry to be familiar, but the critical thing is we make decisions, and those decisions are based upon models, whether they're a mathematical model that's been curated by 6 experts, tested, validated against data, validated against hundreds of data sets in literature, or whether it's a very experienced, technically adept person trying to integrate petabytes of data in their head. The brain's pretty bad at integrating multi-state differential equations quantitatively. It's very good at looking at patterns, innovating, finding new ways of looking at data when they have mathematical support. The computer's good at giving that mathematical support. So that's kind of what I've been doing for the past 20 years. In fact, I've been reading Professor Cabelli's and Professor Dalaman's papers for a great portion of that. My name is Jim Bosley. I'm Senior Vice President of Modeling and Simulation for a French company called Nova Discovery. I live in Philadelphia, so I'm one of the US branch here, and I'll be talking about our platform called GINCO. GINCO is a Japanese word that means both artificial and population. So virtual population in Japanese is GINCO. So I have to look at how models are applied in an industry, because I've been selling, I've been applying these models, I've been discussing these with regulators for 20 years. And I come down to this. One can either apply models to help with decision analytics, which are go, no-go, target selection, and indeed regulatory decisions. Typically, the high return for the decision analytics is early on.
Sorry, Jim.
Yeah.
Just to tell you that you're not going forward with the slides.
Okay.
Okay.
Let me just.
Okay.
Can you see that now?
Yes.
Okay. Thank you. The 2 applications of models are decision analytics, which are to help inform a go, no-go decision, or a yes, no, or a selection of a target. And again, that early on is where that has a huge impact. Later, when you have clinical trial data, you can do very good advanced pharmacometrics. So the incremental add of a complex QSP model may not be super high if your data are very unambiguous and you have exactly the population that you are that is consistent with your application. Later on, we're doing trial design and optimization, and as Dr. Musamba said, the dosing decisions, inclusion-exclusion criteria to optimize the dose. The question is, wouldn't it make sense to have a platform quantitative model to do the quantitative calculations to support those decisions in a better way? And often we, again, the previous talk was very topical. Often you're focusing on a specific research question. So in a project I participated in, the question was asked, how do GPR119 agonists compare to current standards of care or other treatments, so sitagliptin, for example? And we created a model. We leveraged limited published competitor data in addition to hundreds and hundreds of papers in the literature, and we were able to predict within about 5% what the reduction in HbA1c was. This 5% figure is derived from comparison with a subsequent competitor trial, where the competitor did not use such a model to answer the question. And again, the competitor trial both confirmed the model and validated the decision. But the model wasn't used again by Pfizer, to my knowledge. And even though Tristan Maurer told me that the project contributed more value than any other CRO, and yes, I am bragging, I'm going to have that phrase engraved on my tombstone. So in any case, the details have been published, so I'm not revealing anything here. It's a previous employer of mine. And when we talk about, let's say we're building a platform, how do we go about it? Professor Dalaman's talk emphasized fitting individual components, and that's exactly how I approach things too. So I was encouraged by her talk. But we can create these modules that could be used in, for example, diabetes. Obviously, glucose and insulin is there, but we could use those same modules and add a triglyceride module. In fact, I would go the other way around. I would say that triglyceride is nice to have in your diabetes model because there is competition for energy substrate, and you can get that very accurately with by adding the triglyceride model. But you can use these modules in other different areas. So, for example, Nova Discovery has a non-alcoholic steatohepatitis model in addition to some of the glucose insulin models that we have. So, wouldn't it be nice to have all these submodels that are testable in the way that Professor Dalaman outlined? There's a lot of metabolic models that are available, and I'm highlighting some of these here, but they have different strengths, different weaknesses, different inclusions, different exclusions. Wouldn't it be nice to be able to integrate all of those into a model that is valid over a range of different diseases and timescales and therapy approaches? So I'm showing you here a picture of, it's a conceptual picture. However, there are components in this that, well, this entire model is essentially included in the Nova NASH model, but in a project I did with Regeneron, that's the Caxar-Torogi citation that I gave earlier, we used a model by Adrian Pratt et al., and that's the cholesterol input here. And this is a simplistic approach, but it works pretty well. Very simplistic insulin model that can be improved. It has been. And then a triglyceride model. So you can see that we're including both of those, and then we address the competition for energy substrates here. So you can get a model that is consistent with a lot of different data sets if you include all of the different components. So again, carbohydrates in green, the center yellow is triglycerides, the blue here is protein. Then we have cholesterol and bile acids included, and these are representations of models by Macaulay and Morgan. But wouldn't it be good to have a comprehensive, fully tested, all modules tested platform for doing diabetes research? I would argue that a model implemented in a platform does make sense because it has broad applicability. It's a permanent evolving scientific tool. And I'm going to emphasize a component of our platform I think you'll find interesting, which is the knowledge model. But in addition, there's computational models, there's validation, there's verification, there's simulation efficiencies as well. Again, it's a durable competitive advantage for commercial terms, but also, I mean, if you want to look at academic programs as competing and trying to generate the best papers most quickly, it's also a competitive advantage in academia. It has a library of reusable submodels that can be used and tested with confidence in other diseases, and it's a way to integrate publicly related models. So I mentioned that we have a program called GINCO. We've been doing professional services for about 12 years, and a lot of the ideas we came up with, we decided to put into a software platform called GINCO, and then the GINCO is a container for a lot of modeling components that can be used, that we've used for clients. So for those of you that are understand the software business, this is a software as a service and a content as a service business model, in addition to the professional or managed services model. The progress through the models is very similar to what you are familiar with. Again, I'm going to emphasize our knowledge model, because this is something, in previous engagements, I would take 100 or 200 PDFs and put them in a zip file and send them off to the client, and basically they double check to make sure they had subscriptions for all those, and then that would be the documentation. And I'm kind of embarrassed about that. We do a better job. So in GINCO, what you see here is a markdown document that includes a lot of different things here. You know what? I'm not getting a GIF action here. I'll just describe this then. What you're seeing here is a markdown document that has, it's written up by our modelers, but includes footnotes. And those footnotes are references to sections of PDFs. So our methodology is thus. We have 2, 3, 4, 5 modelers working on a project. They go in and this could be postdocs or graduate students in an academic setting if you wanted. And they go into the literature, they tear into it, they go through a PDF, and they highlight key things in that paper, things that must be in the model. We call those things assertions. And those things are noted in our software platform. You can always go back to the source document, but the assertion is something you can cite in a markdown document. You know, kind of old school, you think of making 3x5 cards or index cards where you'd write out a summary of a paper and keep them in a little file box. This is the update of that, and there's some advantages. With this document, when you put the document together with all of your assertions, you now have documentation of the model before you even start modeling. Further, one can take that and send that model or give access to that model to key opinion leaders or regulators or others and say, what do you think of this? Did we get everything? Are we on the right path? What are we missing in the physiology? What are we missing in the data sets we're using? And so we end up with a knowledge model before we've even written a line of code. And that is available to academic groups, by the way. Here's again, I'm not getting the GIF action in this setup. I'm sorry for that. I guess I'm using a local rendition of the thing. In any case, you can see in here each of these things is listed with a reference, and one can click on the little block box and go directly to the citation. There's a number next to that, and I don't know if you can see that, but it's 1.8 and so forth. There's different levels of confidence in the model. So we're evaluating our confidence in the source of each of the assertions right off the bat, and that's factored into our modeling approach. The model that we end up with we end up doing a modular model when we do the computational model. There's a very strong, confident mapping of the assertions directly to the model, and therefore we're sure we don't miss anything. And we also do this in a modular fashion, so we can do tests modularly. And the other nice thing about the system is when we compile the computational model, it generates a list of human-readable, set in LaTeX, if you will, human-readable equations that exactly match the model. Dr. Musamba mentioned validation, verification. VV40 is a very good document. We agree. This is basically how we can generate. There are modules in the platform that allow us to efficiently and effectively and with great certainty generate virtual patients based upon our data sets. And this is an overview, so I'm going to go zip through that, but we can discuss that in detail with folks that are interested. And then last, the platform was really designed to make it easy, once the model is in the platform and developed and tested, make it easy for example, clinicians to do their own simulations. So it's extremely easy to set up a clinical trial in this platform. And it takes, all it takes is expertise in clinical practice, and it seems, our tests seem to indicate it's very straightforward to non-modeling clinicians who just need a bit of coaching, and they can generate their own simulations and generate their own results. And last, the results are easily visualized using sets of pre-coded scripts that will allow one to get a very good idea of what the simulation in the model does. This right here is not diabetes. This is actually tumor radius for a non-small cell lung cancer example, but you can see where we have confidence intervals for treated and untreated arms, which are pretty specifically different. Okay, and as I say, it's easy to design your own protocols. One of the things, one of our founders, JP Boissel, is a fairly eminent pharmacometrician, emeritus professor of medicine from Claude Bernard University, and he came up with what's called the effect model. This graph just shows you on the lower, on the x-axis, the likely response of a clinical outcome in an untreated state, or in a standard of care state, if you will, and the vertical bar is in the treated state. So the further down from the horizontal line, the 45-degree line you are, the higher you are in terms of response. Very easy to pick out responders versus non-responders and identify subgroups that would preferentially react positively to therapy. So, in summary, the comprehensive platform model really does support diabetes in a way that's, I think, much better, much easier, and much more certain for both industry representatives, but also I believe it can support better regulatory decisions. And I would say that I really liked Dr. Musamba's talk. In the FDA, we've had success with, for example, having them say, yeah, you don't need to do a phase 2, you can lower the phase 3 from 3,000 to 700 patients at the cost of continuing monitoring in phase 4, which was fine with, was very good for the client. So we have had regulatory impact in some cases not so much. So I think that there's not an inconsistency between different groups within the FDA. I'm hoping the EMA is a lot better than that. I think you can read the key thing, but I think one of the biggest take-home message for us here is this is something, especially if you're in industry, in academia, we'd like to reach out, and we're rolling out the product formally in early June. So if there's academics that are interested, please feel free to contact me. And that ends my presentation. Thank you for your attention.
Many thanks. That with the last presentation, I guess that we can start the discussion. If you don't mind, Professor Garatini, I have a question to the speakers. Perhaps it's a 1 million question, I don't know, but I'm wondering from the NHS perspective, National Health System perspective, from the societal perspective, what do you see that in the short term would be the realistic benefit obtainable that with the uptake of the in silico methodologies in the drug development and in the drug usage in general. And the other question, perhaps always to Professor Cobelli, you mentioned the legislation and differences between, even across Europe. And so perhaps what are the main barriers that you feel that could be, that we need to consider and work on together?
As far as the first question is concerned, I'm not sure I'm the right person to ask that question. Also, I don't think that there is that much research in the drug area in Italy. So I think that this is all the big drug companies went away. Okay, so there is no real strong backbone here in Italy in terms of drug research. So probably the In Silico Medicine could help other aspects unless things change. The second question is, I wanted that you ask me this question to say, I don't know. Because I'm not a legislator, I'm a scientist that experience dramatic penalties due to the current legislation. So this is really a barrier, bureaucratic barrier. As I told you, we had to return $200,000 to NIH. They went to France, where they had this opportunity. In 1 month, they have this tool that I mentioned, similar to the FDA, in order to do experiment. If I have a sensor, a pump, and an algorithm, the ministry does not give me the permission to do an early feasibility study. But forget myself. Think on the small companies, not big companies that have a lot of money to invest. Small companies, innovative, radically innovative, that cannot do this kind of studies. So here we must call for legislator.
I have also a question that maybe is easier to answer. I haven't seen any mention of gender difference. Have you considered that aspect in your models, or?
Yes, we have. There is a paper 2006, gender. Sorry. There is a paper in 2006 in diabetes, gender and age differences in glucose homeostasis. So there are differences.
But I mean, in your algorithm that you are made up in order to establish the model, has this been taken into consideration, or?
It is there because the data were coming from different populations. So I think it is there.
Well, but if you don't establish different algorithms for male and females, it is very difficult to say that is the same or that the data are there.
Okay.
We know it from many other aspects of the literature that we, in reality, are punishing the females because most of the data are established in males.
I think your point is well taken. From the big studies that have been done on artificial pancreas, so we are speaking now of almost more than 1,000 subjects with different gender, different ages, etc. So the algorithm was not tuned to the gender because it's already very difficult to control the system, with...
Because in most of the studies, all the phase 3 studies, females are present in a very small percentage, and therefore it's difficult at the end to establish if there is any difference. The data are made without taking in consideration possible difference. That's why it.
No, I think your point
I believe
is well
that
taken.
this is a point to be considered.
I think so, and now, when you submit a paper, usually people are very careful on the gender balance.
Well, not anymore. Not at the present, I believe, because in my opinion, there should be, for clinical trials, there should be a different protocol for males and females, because even in the calculation of the sample size, the incidence, the prevalence is different. The onset of the disease and the course of the disease is frequently different. The outcome is different in females and males. And that's, I believe, it's an area where there is a lot of work for you.
Okay, that's a good suggestion. Thank you.
I try to also go on a more positive note on the answers that you gave, Professor Cobelli. I guess that for sure there are hurdles from the legislation, and it is surely true. But my question was also what you foresee that we can take on with this in silico methodologies applied to drug development, to speed the development, to come earlier to patients. And my question was, what is, for instance, I see that in some cases rare diseases are something that in which we could move forward, and this was the kind examples of the rare but not so rare patients. That was what I tried to refer to. I mean, just to be focused on what is something that we realistically can achieve in the best interest of the society. I mean, by the uptake of this. But perhaps I wasn't fully clear in my question. And I had also a question that I would like to take from the public that is following we talk about the virtual trial and the benefit of virtual patients, and there is an interesting question, because it seems that the data that you can have is mainly focused on the benefit, and there's a question of how this virtual trial can reconcile with the need to collect safety, in what terms it is something that it might be helpful, perhaps it worth to clarify to the people that is listening to us. I don't know who wants to get.
Thank you. That's a very good question. And I believe this exposes one of the main limits of modeling and simulation. Models predict what we know. They struggle to predict what we don't really know, okay? So there is a lot of application of modeling and simulation in safety, but the most effective application are just to test known failure scenario. Or known adverse effect, okay? A classic example, for example, is the in silico cardiotoxicity. There are some very well-known mechanisms through which a wide class of drugs interfere with the QR phase. These are well-known, well-described, and there are various groups around the world that have implemented sophisticated in silico studies to test any new molecule in terms of cardiotoxicity. What modeling falls short is the unexpected, completely unexpected complication. However, it is also fair to say that even a clinical trial is a model of the reality. So if we don't look in a certain direction, we will not see, and things will appear only in post-marketing. So Professor Garattini knows this much better than me. So it's a complicated argument, but yes, it's an important point.
May I make a comment?
Yes, please.
This is Jim Bosley again. And if, in case I forgot, thanks to all for inviting me. I really enjoyed being with you and hearing the talks. As usual, Professor Viccicanti is spot on. I'm going to give a little more hopeful, and I hope not too Pollyannish response. When one is modeling, one has data sets, one fits the data sets, one gets a good fit, and then one compares with data sets one didn't use, and you get a validation, and you've gone forward. Sometimes something happens either in the efficacy or in the safety arena that you didn't expect. And the model doesn't capture it. And you know, we've been putting our heart and soul into making the model match reality, and it doesn't, and people tend to look at that as a failure. It's not. For exactly the same reason that Professor Viccicanti mentioned, the clinical trial is a model of reality too. There are things in clinical trial that you didn't expect as well. A mismatch between the model and the data set in a well-made model that's mimicking a lot of data, a mismatch between new data and that data set is an opportunity to learn something that nobody else has ever learned. It's an opportunity to expand our thinking and to eventually correct the model so that it incorporates that. So I view those things that you don't catch initially, or model mismatches in safety or in efficacy, as an opportunity. Of course, I have had clients that have asked me to model idiopathic safety issues, and that's that's a little different. Thank you.
Okay, thank you.
Can I follow?
Yes, please.
Yes, thank you. That one is a very interesting one. I just want to also comment in line with what has been said, but I think that this is a clear illustration of what I was trying to say here, saying that you have no model, and I like a lot the analogy from Marco, saying that clinical trials are models. Yes, there is no model in the larger conception of that cover everything. Each of the model tackle selected particular question. You cannot have something which is not now perfectly applicable for everything. And in order to have a good model, being it in silico model, you need to design data collection in the very start in such a way that the model is fed and is then able to predict the next situation. So it is possible that you have very good model tackling the safety issues. The problem is that for now, no one invests into developing this kind of models, into collecting the kind of data that would lead to having confidence in predicting safety. Yes, because it's more challenging. Yes, because it will demand much more investment, much more data whatsoever. But theoretically, it's not impossible. But you should not ask a model which was initially conceived for addressing efficacy-related questions to also make good job for safety. No, there is no magic. If you want to predict safety, you need to kind of concept your model in order for it to be able to address safety-related questions, and that's why there are some of those. We have very good model for cytokine releasing syndrome, really validated and doing very good jobs. So if there are questions that modelers think could be interesting, the whole exercise is about having credible, validated, qualifying model to address those. In theory, you can have model for all the questions, but we cannot use a model initially developed for one question and sort of expand it to cover all kinds of issues that we can have. This is just not gonna work. Model is a method for questions that need to be predefined from the very start. Thank you.
Okay.
Okay.
Thanks, Flora. I guess that we are going to end this meeting. I guess that to go back to where we started, I guess that it's pretty clear that we need to talk each other much more. And because we have different perspectives, and we heard here today with many potentials and many really amazing things that are potentially game changer and something that on which we should align, and we need to have a common understanding of how to best use this one. But yes.
Yes, no, I agree with you. And in addition, I would say that it's very important that all these topics enters in the School of Medicine, because they are still out of the contest in the medical education. And if we don't do it very rapidly, we will have a very large gap between what is needed and what is learned. So I believe that this is another type of, let's say, task that we have. We cannot keep this area completely isolated from the rest of the medicine, where it is very important that it should be applied. And in addition, I would say that it's also very important that this approach is utilized not only for the benefits of drugs, but it should be utilized also to establish the best condition to prevent the diseases. Type 2 diabetes is an avoidable disease. It can be avoided probably by more than 60%. And if we are able to establish models that we can propose to people, I think we will achieve a very important goal. And I believe that it's time for
yes,
Dr. Magrini
yes.
Professor Magrini to...
Yes, I kindly ask to our Director General, Nicola Magrini, which I thanks for the support for organizing this meeting. Please take my seat.
So, gracias. So, thank you, everyone. I listened only partly to the second part of the meeting. I found some interesting elements. The idea that we promoted this contact and exchange seminar is to try to have a more common language in how do we arrive to best have the best available data for first in human studies or modeling and predicting how it will function in human. I think it's still an early dialogue, I would say. I'm currently daily fighting for another objective, which I think is still very far. Why is the evidence for approval so different from the evidence we would like to have for reimbursement and for best use in the National Health Service? So the idea that you propose to have model that could help early in the phases of better design of the studies, that is absolutely fantastic and a common objective. For this, I'm here also to say we can commit as IFA, though some of the approvals do not depend on us or some of the legislation is far from perfect. I think if we join our effort in promoting some of these research for feasibility, even when the law apparently is not easy to interpret, I can say if we commit together and or facilitate tables to allow better human studies, we usually enter also with the ethics committee when the phase 1 is ready. Here at the interface, I think we can do, if not a lot, certainly something more and something more together. I heard some interesting discussion on how much even clinical trial and randomized clinical trials are a model. Well, certainly they are, but the larger and the better they are designed, the less they are a model, the more they are a good model to be sure of the estimates and to predict pretty well the effect. So it is true, it's always necessary to keep validating the model in all forms. This is what research is there for. And some discussion also on what kind of clinical studies or clinical trial do we need is always good to have in all communities. So let's take this as a first entry point or a first workshop. I think some of the backstage discussion we had were even more positive, showing a common language and a common attitude. Today you went into some detail or some more in-depth discussion, and sorry again for being here only on the second part of the meeting. If we can plan for more problem-oriented or case study-oriented examples where we can play a more active role, the better it is for IFA and for the role IFA can play on or in independent research promotion and support. We have a little fund for that, and if there are gray areas where we think it is of NHS relevance to promote independent research, we can certainly promote a discussion. And not sure we can now commit to definitely fund it, but certainly open the door for a discussion. If it is of National Health Service relevance, we can be there for that. Silvio Grattini, I thank him for being here and for having promoted the independent research fund for Italy, which is a law we are quite proud of, or at least I am. On the issue of gender differences, I think it is now time to be more accurate on that. I'm not sure, I perfectly understood what Silvio was saying on need for different sample size, typically for diseases that have different incidence in male and female. Not sure the solution have to have two different protocols. I think we start from there when we add the protocol, the famous protocol for S studies in male only to demonstrate statin effectiveness in myocardial infarction. I am old enough to remember the publication of that study that was perceived as excellent, all fine, we are sure for men, it will be the same for women. That's how it was accepted. And I'm not sure going back to that and promote two independent studies, or unless we have the capacity to be binding. And if we are binding and promote always two parallel studies in male and female, well, that could be an interesting revolution, but I'm not sure it's time for revolution this decade we are living in. But post-COVID, probably everything is possible to discuss what we didn't dare to discuss before. Not sure that with the war we are now going in the right direction, including for research and support. I think this is, for me, is a very painful situation. I can confess I am clearly against this spending on weapons and so forth. I know I shouldn't say so because I am on an institution and say, but I think it's institutional what I'm saying. I'm very worried. Funding for research and funding for the NHS could drop in a few years due to too much money spent on weapons and so forth. So now I certainly expanded to an area, and moving back to whatever can be now a good basis to discuss potentially big differences between female and male. This could apply to this kind of modeling. I think this would be an easy convergence of interest. And again, if there are either simple or complex case studies on what we could promote as potentially useful for better research, better clinical studies, for the development phase, this is something we can commit to. It's part of our role. And also facilitating when the hurdles and obstacles you mentioned are not rational or are old and should be changed. I cannot say we can change easily the law. This is probably the most difficult thing I've seen in Rome. So I'm not promising any legislative change easily, but probably finding possible solution together, putting the right people or all the people around the table could be something we are open for, and probably this morning can be seen as useful if we promote more dialogue and more regular exchanges. So with this complete willingness to be ready for regular, at least twice a year meeting, for example, we could promote more and more specific discussions on studies or issues relevant for research and relevant for public health. That is the main objective of our work. So thank you all, and thank you also for those who participated from distant. Thank you.